Hello, it's February 4th. It's day 2 or attempt number 2 at the personal human genome sprat mapping, what I'm doing here. Having fun with the human genome. Today I want to make a stride every single day. This is the beauty of consistency. Every single day I want to do something to get my genome in Excel or somebody's genome in Excel and start analyzing it with pivot tables. Made some progress over the last few days. Really warm, receptive. Most people hate my videos. Everyone seems to like this video. I guess people are interested in organizing genetic data and perhaps extending their lives indefinitely by synthetically creating DNA just the way you need it to do whatever you need it to do in part of your body and make it work right. Funny that people might be interested in that. Anyway, seriously, overwhelming response has been awesome. 4 likes is the most likes any of my videos has ever had. And people like Mr. Excel, Bill Jelen, the man himself, chilling with Jelen. He is an animal when it comes to Excel and knows everything. He's been helping out, giving me bad news. He thinks it's bad news, but it's not bad news. We're going to solve these problems and get all this data in here. And so I want to tell you what I've done for day number 2 in the update. I quit my job to do this kind of stuff, and so I'll tell you what I've been doing. Now, first of all, I need to get a hold of my genome, right? I mean, without my genome, it's not going to work. I'm using somebody's genome mapped in 2003. This is what you're looking at on the screen right here. Look at this mess. Does this look like gibberish to you? TACIGAGNNN, all stuff that's all over the place in a complete mess. It comes in 70 character rows, and I just went to the bottom to row 1,480,000 or whatever, over a million, deep in there, and this is a complete disaster, right? What I did to turn these 70 character cells into a linear string, a sequenced linear string of bases, was I wrote some funky formula using some simple formulas, arguments or commands that Excel has, and I said, I think I might have oh yeah, here we go. Space is mid count. I basically went in and I said that we want to have, because all of these columns, or all of these characters in this column are 70 characters long, like every one of these is 70, even though this looks longer down here, that's because the character N is wider than C's and A's and T's, and that's why it's stretching out to the right more. Those all have exactly 70 characters in there, at least they should. I did a check for that early. So what I did was I created a 70 row long character string. You'll see this ends at 70 right here. And every time it goes past 70, it restarts at 1. And I use this as like an anchor to pull the proper base pair or nucleotide out of here and put it in order. That's what this formula does. It is it's a mid VLOOKUP. I write formulas and then I forget what I did. The mid thing here says go into look at the row ID. Oh yeah, look at the row ID too. We have to ID all these rows. That's what this column D is. You'll see that goes down 2, 3, 4. Every 70, that row ID number goes up by 1 because it then helps to look back to the right cell. But it basically says, I'm not going to explain it because it's too complicated for me to explain. I forgot what I wrote. But it pulls one character at a time. It pulls it and then creates a line. Now, what will happen is, you see that I've got a full, when I imported this off of the National Institutes of Health website where it's public, look, it goes all the way down to the bottom. I'm going all the way down to the bottom to row 1,048,000. I didn't get that to be able to turn that into a vertical character string because it's too long. Basically, think about it. Every one of those is 70 rows down. Every one of these cells with 70 characters is 70, converts into 70 rows down. So I did not get there. I only got part of it, and you can tell how far I got by right here, this row ID number. This says basically I got to row 14,981, and then it ran out of space here at the bottom. And we're going to remedy these problems. I've been talking with a few people who have some Excel knowledge, including Mr. Excel, who's keyed me into Power Pivot, which is a software program that's an add-on to Excel that allows you to do big numbers like billions of records. You can get them in there. Writing these formulas next to them, however, is an interesting quandary that we are in, because however Power Pivot works under a software language called DAX, which I'm not familiar with, it's DAX, you can get these records all in one thing, but can you make them talk to each other based on what order they're in and stuff like that? That's stuff we haven't been able to figure out the answer to yet, but I'm sure we will. Now, 14,981, which I'll get back to, that's a row, and we got there. Unfortunately, we that's as far as we got. So this is the like 45th character from row 14981, and if we go up there, I'm sure there's a faster way. Freezing for a little while. Come back to the computer. So there's a ton of records. I think it's about 27% of chromosome 1 is listed in here in this column. Yet far less than that. Really only 1 million of the possible 250 million characters are in the sequence, which I have brought over to this other sheet where the data is, where I've been working with. These are all your bases, right? Up down to 1,048,000 of a possible 250 million. So whatever, a third of a percent or whatever that is. Small amount, right? Now, I've done some formulas here to have some fun and to be able to look for patterns and have patterns in my pivot table. So if you go back to what the pivot table is showing right now, I started looking for a 4-character pattern called TAAC because that's the first one I saw, which apparently is the telomeres. These telomeres are these 6-character TAACCC that appear at the beginning of the ends of the chromosomes. You see at the beginning of chromosome 1 here, it's probably at the end too, which we don't even have because we weren't able to get all the data in here yet. So I started setting up a little formula. I wrote something using named ranges, which just basically identifies, like this cell is called _A. I named this cell _A. And so anytime I change this, if I change this T to an A, press enter, all of a sudden you'll see once this calculates, this is going to change. This is looking for a 4-character pattern of AAAC, right? You'll see it's processing it. Now all of a sudden it went false here because this is not AAAC, it's TAAC. So if I go back here and I make this thing T, and wait a second, as it processes through the million rows, you're going to see that thing turn to true again. Now, the point of doing this, you could do this with a 4-character, with a 3-character, with a 2-character, with a 5-character, with an infinite number of characters, not infinite, but you could do it with a 1,000,000-character long string here in Excel, and you can then use that to determine, what does this pattern mean? Does this pattern create a specific type of protein? Does this pattern tell the cell to do something else when there's transcription and RNA takes it over to other places and it does other things? It's very complicated where how this DNA software coding relates to cell structure and interaction with the entire body. There are many levels and dimensions of understanding that we have to gain to get that. But it seems to start here, or at least this is the most, broken down way we can look at it. See, there we go, boom, we got the true. Now, this table over here is looking for this TAAC, and it's showing this by 100,000 base pairs of order, meaning this is like the first probably 50,000 characters. This is a chunk that's looking at the next 100,000, then the next 100,000, all the way down to, if you do 100,000 times 10, you get 1,000,000. So that's why we have a little more than 10 slices here. And what you can see is the TAAC pattern actually appears the most within the 700,000 rung of this 1,000,000-character data set. So you can start to graph these relationships and find them. That's what I did here. And what else you can do is you can kind of open up and you can see the number of each nucleotide that exists, which manual calculate on, which means the file won't blow up on me every time I try to do something. It'll only blow up on me when I ask it to, which is much nicer. It has to do with going into File and then Options. Never done this, and then you go to the formulas right here and you click this for manual so that stuff doesn't calculate automatically when you have a billion formulas going on like I got going on in this file right now. So what I was looking for, was messing around with, I'm just going to make it a little easier because I want to show what I want to show. This is the distribution of this 4-character formula of TAAC right here. Well, I don't want to look for TAAC anymore. I want to look for well, no, not that, GCCG. Why not? This is a lot of cytosine and guanine, I guess, which happen to bond together, so it's kind of the same thing. I'm going to look for this 4, and now I am going to have to calculate. Please don't blow up. I'm going to do that, and once this thing changes to false, because I know it is, then I know the whole thing will be done. And it is. Now I go back to the pivot. What we were looking at, I haven't refreshed this pivot table yet, but what we were looking at was TAAC, I think. Now we're looking for the existence of GCCG for whatever reason. And when I write, man, I mean, this is in blue, so this is going to change dramatically. Boom! Look at that. So now this is interesting. It's occurring a lot compared to, you know, if you map this with data bars to see what this actually looks like numbers wise. Use some conditional formatting. Do some data bars here for colors for fun. You can, you know, it's similar to the chart actually in that sense, but you get to see how much more, oh wow, this is something that apparently starts to occur 800,000, 900,000 into the 1,000,000th row. More so, it really doesn't exist at all here. So if this 4-character thing means something, and you can start trying all different 4-character combinations and 3-character combinations and 5-character, and all this, it's gonna, you're gonna be able to graph this and table this visually and really get a good idea of what's going on. I'd like to show what this looks like without just the trues, meaning showing the falses as well. Because what happens here when you do this, this graph is going to look a little screwier. It's not really going to make a lot of sense. But what happens when you open this up is you start to see, remember we knew from before that, you know, most of the trues are in here. You open this up and you're going to see what each base pair and the number of times that it exists. And of course this starts at G, so G is the only one where it's true, but there's actually all these, wherever it's GCCG, that's actually 4 trues. So some of these C's and another one of these G's, 471 of these G's are actually should be in the true column, and I'll show you how to change that actually later. But this then shows a distribution of what's ACCTG in all of this area as well. I'll do some more conditional formatting bars and show for like this. You can see that in this instance you do see a lot more C's than you see A's, right? And this A's and T's. Now what happens when we open up, you know, compared to, I'm going to use slicers to do this. I'm going to create insert a slicer for the 100,000 character groups that I've created. So it's going to be basically 0 through 10 here. And what this does is this shows, this is going to show, I like a lot of colors. This is going to show, when I click on a couple and expand, it's going to show a comparison between ones that I won't looking at. For example, in the 300,000 range, there's only 2 instances of this GCCG, right? I want to compare this 300,000 range to this 900,000 range because there's a lot of the GCCGs in this one, and this, you know, this is the most. So all I do is I hold down Control and I hit 3 because I want to look at 3. Actually, I just hit 3, sorry, and then I hold down Control, and then I also hit, because I'm holding on Control, now I hit 9. And now I have both of these. And I can open them up, and I can do many different things. I can do, you know, more ratios of what's to what here by doing conditional formatting bars. Actually, the graph is going to be really helpful for this too, right? Yeah, we should just use the graph. Graph is going to show, there's so much that you can do, I don't even know where to start. There's so much going on here. What is it saying? It's saying the reason, yeah, here's the reason why we didn't get any information in the 300,000 group is because there's a bunch of N's, and N's are not good. N's are nucleotides that are unidentified, so this is bad data. So this is what the great thing about pivot tables and slices is, you're able to see exactly what's going on. I know now, after diving down immediately, that perhaps there are more instances of GCCG that exist within these, but because there are so many unidentifieds, you know, how many N's do we have? Where are our N's? Our N's are not base pairs. They are unidentified chisel. Where are they? Where do they exist in our data set? Well, when I create a filter for the bases, which is what I'm doing, and I'm changing colors on it for fun, and I say, well, where are my N's? All I have to do is click on N, and what's going to show in the table is going to be, there's where my N's are. Oh yeah, my N's are in the middle of this first million in chromosome 1. Right? They are right here. They're in 3, 4, and 5. And how many of those 100,000 are N's? A whole freaking lot of them. Almost all of them here. Craig Ventnor, you dirty prick. Where is your... I hope our genome sequencing has gotten better than this, because N's are not going to help me. Question marks are not going to help me. But these pivot tables are. So you can see the fun I'm starting to have with this. I have stolen my wife's credit card and purchased a genome on 23andme.com, and they have a developer area there as well. I'm sure I'm going to contact them. I actually chose the option of pick up my kit, where I'm going to swipe my DNA on the inside of my mouth, pick it up. You can get it mailed to you, express mailed or whatever. I don't know where I have to go to pick it up. It could be New York, it could be San Francisco, it could be Hawaii. But wherever it is, I'm going to go get it, and I'm going to talk to these people and show them what's going on. Because this is day 2 of the human genome, and I've only got like 300 something to go. So let's keep it up. And that's Ken's Talk, day 2. Genome it up.